Just wanted to announce there's a seminar at 1 over in the Mead Auditorium, which is kind of kitty corner on the first floor in this building. So Ella is going to be giving her presentation on her master's thesis project, looking at witches' brooms and caribou grazing. And witches' broom is actually a disease. There's several different pathogens that can cause that. This is a fungus that's causing this impact where the fir trees sprout these little scraggly looking broom-like growths of their branches and twigs and leaves. And so I was trying to find a little bit more in depth on like how does the fungus actually affect the plant to cause this, and there's not a whole lot done on it. My guess is there's quite a bit of hormone shifting going on where either the fungus is exposing that portion of the branch to a lot of like gibberellins and auxins, or stimulates the plant to do that in some way. But anyways, she's going to be looking, she did her work on the Slate Islands in Lake Superior, which is a pretty interesting way to look at some of our ecological questions. So should be a good presentation if you want to go to it and see what a master's thesis seminar is like. I encourage that. All right. So I think we left off here with the idea that you can have, these secondary metabolites, glucosinolates. And this is another idea of the chemicals in the plant and the enzymes. They're spatially separated, so this reaction isn't going on in the plant until it starts to get chewed on. And then it breaks down the compartments and membranes and allows for these chemicals and enzymes to interact. And you can get the isothiocyanate compounds that are secondary metabolites trying to defend the plant against the chewing herbivore. So this is just another look at that, where you have both cases here, both examples where in the plant it's spatially separated, but upon herbivory, the chemicals and enzymes interact, and you can get some pretty potent secondary metabolites that the plant's trying to use to defend themselves. So with the, let's see. So those, okay, nitrogen containing compounds. So we've covered everything except I wanted to get into this idea, the non-protein amino acids. So these are part of the nitrogen containing compounds that serve as secondary metabolites. So these are amino acids that don't get involved in protein structure. They're not part of that. And so they're not incorporated, they're amino acids that are not incorporated into proteins, but they look very similar to the amino acids that are used for protein synthesis and part of the structure. And so basically we're thinking of them as a mimic amino acid. Canavanine is being shown on the bottom there, and then comparing it to arginine, which is used in protein synthesis. So you can see that very similar. And so what happens is if a herbivore ingests some of the canavanine that's being made by the plant, and it can act as a mimic to arginine, and so then it'll get incorporated into the amino acid or into the protein by mistake, so to speak. It's just mimicking, and then that can cause that protein, once it's all put together, it won't function correctly. Often it doesn't get folded in its correct three-dimensional shape to allow for its normal activity. And if you've taken BI-218, you know that, proteins, yes, there's strings of amino acids, but then they have to fold and twist into a three-dimensional shape. And that shape is critical to how that protein functions. And so if you mess with that, you're going to mess with that protein. And then, of course, the organism who has messed up proteins is going to suffer for that. And because of that. So that's an idea that they've seen, that some plants will have a repertoire of these non-protein amino acids to serve that function. Then there's some specific proteins that the plants can make that will mess with the herbivores' digestive enzymes, essentially. And so alpha amylase is a digestive enzyme. We all have that too. That's in our saliva in pretty high quantities to break down starch. And so a lot of herbivores are needing to do that. And so lectins are proteins that plants have. We already talked about lectins way back when we were looking at how they bind the signal from the nodulation factors will bind into the lectin proteins from the nitrogen fixing bacteria. So the lectins are part of a normal kind of thing that plants have, and they can bind to carbohydrates. Well, if an herbivore ingests some of these lectin proteins by the plant, then these lectins can bind with the carbohydrates as well in the herbivore, and it can bind to the digestive tract of the insect, the herbivore. Often it's an insect that they've studied. And so basically what ends up happening, if they get enough of that in, especially in a high dose, it can interfere with the ability of the herbivore to digest. You may have heard a little bit about lectins in the popular media. There's a lot of hullabaloo about lectins in our diet. Kind of thinking along this line, is it doing this to us? And so for a while there's a lot of experts, and I'm putting the air quotes in there for purpose, the experts saying, oh, terrible, terrible, don't eat any lectins, don't eat any foods that have lectins. And then you start looking at their list of what these foods are, and a lot of them are really healthy for you. Just saying, don't eat any of these. And so that's kind of a pretty broad brushstroke statement. And you always should kind of have a red flag up when you see that kind of massive, broad statement. And so now I followed some of those studies where they're doing more careful research and finding that that's not necessarily true. Lectins actually can have a beneficial influence into your health. And so basically, at least the published studies I've read, they're saying that lectins are not evil and that it's okay to eat these foods. And often the way we eat these foods is we cook them, and cooking them actually does reduce lectin levels considerably. And so if you look at how we naturally interact with these foods, most of the time we're not even getting exposed to high levels of lectins anyways. And so whenever you start hearing these, blanket huge statements of all of these things are evil, you should do your own research and figure out if that's really true, or if they're just trying to make a quick buck off of that. And usually if you follow the money, you do see that indeed they are trying to sell a product that will help you deal with lectins or whatever it is at the moment. So be careful about that. Protease inhibitors are another idea here, where the plant can make their protease inhibitors. The herbivore, chews on the plant, gets exposure to that, and it again will mess with the herbivore's digestive enzymes. So proteases break down proteins that are digestive enzymes. And so again, you're messing with those. So many different strategies. And again, it all kind of goes back to the idea that plants can't run away from whatever is attacking them. And so they have very, very sophisticated mechanism in place to try to persuade whatever is attacking them to stop or else. So there's a whole book written on this topic. Has anyone seen this one? Wicked Plants. The Weed That Killed Lincoln's Mother and Other Botanical Atrocities. It's a fun read. Amy Stewart has written several books about plants and plant-related topics, and they're all good reads. So she goes into all of the poisonous plants and what that is all about. I can't help but also mention in this slide set, another related topic is the idea that maybe it's not just the plant that's making these secondary metabolites themselves. Maybe they have help. And so plant physiologists I think, were a little dismayed initially when they realized all this time that there are microbes in the plant tissue that are also producing secondary metabolites. And so then they're like, okay, so all the early research that we did before we knew that, now we don't know if that was the plant doing it or was it the endophyte. And so I do want to put in a plug here to get you thinking about this idea. If you go on into a plant-related job where you are extracting secondary metabolites for the company that you work for, you need to consider the idea of endophytes playing into that role. And are they producing the secondary metabolite as well? And if so, that can actually be a really good thing, because then it's way easier to, you can isolate that endophyte and create a situation kind of like with brewing beer. You put them in huge vats, give them all the right nutrients, and they can produce that secondary metabolite for you, usually much cheaper than growing the plant and extracting the secondary metabolites from the plant. So I wanted to just put a plug in here to get you thinking about this. The word endophyte is within the plant. That's what that means, within the plant. And so you're thinking about microbes that are in the plant and not typically causing disease or anything negative necessarily. They're actually, just like with all the microbes that hang out in us on a day-to-day basis, we're finding many of them are doing really good things for us. And we think the same is true for the plant. In fact, there's numerous studies now that have been published showing how endophytes really can help a plant adapt and thrive in certain habitats. So you're looking at a picture of an orchid leaf that has been put down onto tissue culture medium, and you're seeing the fungal hyphae growing out from the tissue, the plant tissue. So this is an endophyte. So look for that when you are looking at your tissue culture vessels tomorrow. Should give them a good look over and see if you do have any contaminants. If you see microbes, bacteria or fungi kind of oozing out from the tissue, like the edges of the tissue, those are probably endophytes. If you just see a massive swarm of microbes in the agar or on the agar, that's usually a contaminant that fell in during your manipulation. So you can usually tell the difference between an endophyte and just something that floated in and landed on the agar. And there's really nothing you can do to try to kill off the endophytes in tissue culture processes other than loading the agar medium, the tissue culture medium, with antibiotics. And then I've tried that. You have to be really careful because some of the antibiotics that tissue culture companies sell, the plants are also sensitive to, and it's toxic to them. So I've had to call and complain a couple of times, and they're like, oh yeah, well yeah, some plants are, you know, really sensitive to that. So sometimes it can be very challenging to find a situation where you are free of endophytes. So we are now realizing that these endophytes are often mutualistic. It's a win-win situation for the plant and the endophyte. And we find that they can be good biocontrol agents. They can inhibit pathogens that might attack the plant, and herbivores chewing on them by the production of certain toxins to the herbivore, for example. And they have been found in numerous studies. Endophytes are critical for certain plants to adapt, especially to harsh habitats. And so the idea is similar to what we're finding in humans. We're all colonized, all of us. We're colonized by our microbiome, all the microbes that live in and on us. Plants are that way too. And so there's been a lot of big research push into understanding this better so that we can harness these endophytes and use them for helping the plant, and also because they produce such interesting secondary metabolites, they can also be used for drug discovery as well. So they actually refer to it as bioprospecting, where they're hunting down endophytes. And if you took BI-230, you learned about that with that documentary by Dr. Gary Strobel, where he went around the world hunting for endophytes and was able to find many endophytes that have really interesting characteristics in biology. And so what we're finding is that some endophytes produce the same secondary metabolite as the plant does. So that probably means they share genes, and often it's the plant, there's genes taken from the plant, so to speak, that the fungus can pick up and use to express the same secondary metabolite. This is probably the most famous example with Taxol, the drug Taxol, and it's a breast cancer drug. So it is one of the most successful breast cancer drug treatments out there. It can put you in total remission. And so it's only effective against very particular types of breast cancer, but you can see its chemical nature. It's pretty complex. They don't know how to make that synthetically in the lab. So initially they found out that Utrees produced it. So Taxol, that's where they got the word from. Taxus is the genus name for the yew tree. And so they started chopping down yew trees left and right. And then Gary Strobel and his colleague said, let's quit chopping down all the yew trees and see if we can figure out a way to do this. What if there's an endophyte in the yew tree that's doing this as well? And so they tested the yew tree for endophytes, and sure enough, they found a fungus that is producing Taxol. And so it's much easier to take that fungus, purify it, grow it in big vats, and get it to spit out this Taxol drug. And all you have to do is purify it right from the broth. It's not that complicated, really. Way more cost effective than growing yew trees, waiting for them to get big enough, and chopping them down and extracting the chemicals. So there's a lot of potential here for finding very interesting secondary metabolites without driving our plants to extinction or creating a lot of extra effort just trying to grow the tree or plant to do the extractions from. They've also found that there's lots of endophytes that produce chemicals that the plant does not, which makes sense if you think about it, because that would be good for the plant to keep that endophyte alive, keep feeding it, so that it's making something novel that the plant doesn't have to bother making. I mean, that does make a lot of strategic sense. And so, again, they're very interested in working and studying these endophytes to see what other secondary metabolites might be out there that could be useful for human and animal health and plant health, of course, too. So there's quite a bit of research going on with this. Gary Strobel's, if you remember that video documentary about how he got into it, it was kind of serendipitous how he fell into this field of research, but then he went with it, and so he's published quite a bit. So here he is with co-author. Is a review paper just looking, he's pretty much advocating that we need to really make a push for this and really better understand these endophytes and study them, see what we can find. And so lots of interesting studies going on and what they're discovering. But again, just looking for, natural products. Essentially, we're looking at, secondary metabolites, and we're looking at all kinds of ideas, antibiotics, antibiotics, cancer compounds, the list goes on and on. And so they're really excited about this idea of, can we start isolating, purifying, and studying them? Here you can see in this next one, this is also kind of in the same realm as we need to understand the importance of endophytes, because they're starting to realize that the seeds that the mother plant makes have their own suite of endophytes, both in the seed and on the seed. And so now they're starting to think, do we really need to surface sterilize our seeds all the time? Maybe we're doing that plant more harm than good, because those could be very important microbes for the plant as it starts to germinate and grow. So now there's a lot of effort looking at that idea. Cannabis work, you can see that they're looking at the cannabis microbiome as well, and looking at the endophyte role. And here, this particular paper is looking at how cannabinoid compounds can be stimulated through symbiotic and or mutualistic relationships with the endophyte. And so very interesting work looking at, how are they upregulating the the cannabinoid pathway, and they're looking at hormone effects, what's going on with that, in looking at the impact of the hormone production by the plant. Here they're saying a mixture of 4 bacterial endophytes significantly improves CBD and THC content. Endophytes could be manipulated, or they could manipulate the ACC deaminase level, the precursor of THC biosynthesis in the plant. So they're looking at how these endophytes are influencing the plant's physiology for some key secondary metabolites. And then this, I couldn't help but put this one in here. It's just showing the complexity of biology. Here's a bacteria that lives inside the fungus that lives inside the plant. And so again, just showing that the more they look, the more they can find in terms of complexity. I'm not going to talk about this. I just thought it was a interesting find that they're able to do this. It's almost like, what are those, the Russian dolls, you know, where they're nested in each other? It's kind of like that idea. And they think that this is necessary, like the bacteria inside the fungus is a good thing. It's actually part of this a dynamic of all these players in the game, that it is a mutualistic situation. So all kinds of things to study with regard to endophytes, but I wanted to put that in this slide set just to get that in your head, that yes, plants are amazing at producing secondary metabolites and we have so much to learn, but endophytes are there too, and we can't ignore them. And actually we need to study them because they could be incredibly useful in so many applications. Okay, so boy, you could go on and on. If those of you who are in chemistry, you know, you could go on and on and on with this topic, but I'm going to stop there and shift gears into, okay, yes, plants make all these really wonderful secondary metabolites, but now how do they use them? And what is the process that they do in order to defend themselves? So as we were saying earlier, most of these secondary metabolites, the reason they're getting made is in response to some kind of stress in their environment, whether it be abiotic or biotic. I'm going to focus right now on the biotic, looking at something that's attacking the plant and how does the plant use these secondary metabolites. So we're going to get into that next. So mechanisms of defense. Okay. So mechanisms of defense. So now we're thinking about all the different organisms that can attack plants. Here you're seeing some caterpillars. Oh my goodness, there's so many insects out there that plants have to deal with, and let alone, and then just think about all the fungi and the bacteria and the viruses. I'm not even going to get into viruses because that field of research is very challenging. Well, here's a nice poem you can read on your own. It's very nice. But so this is the famous disease donut. If you take a plant pathology class, they talk about the disease donut. And so I wanted to include this because it's a broad picture, big picture idea of what's happening. So you have on the left all the abiotic interactions that the plant might have to deal with. On the right, you see all the biotic. So we're going to get onto the right side of it and start talking about some examples. And then after that topic, I will get into some of the abiotic stresses after this topic. So we'll get into both. One thing I wanted to point out is that just like how when we get attacked by pathogens, it's typical that whatever's attacking you can only target certain parts of your body. So for example, when you catch a cold, that virus is adapted to dock into receptors or onto cells in your nose throat area. And that's why a cold virus isn't causing problems in your kidney, for example. It's the same basic idea with plants. They have pathogens that tend to have co-evolved and adapted to particular tissue types of the plant. And there's some exceptions, of course, but that's the general idea. Here you're seeing this poor tomato plant that is, they're trying to show the variety of different pathogens and where they have adapted to attack the plant, what part of the plant body. And so you look up some information on tomato, they're attacked by more than 100 different pathogens. that we know of. And so that's a lot for a tomato plant. So my sons always tease me that I buy organic ketchup. Why are you doing that? And I said, do you realize how much pesticides they have to spray on tomatoes to get lovely tomatoes to show up in the or ketchup? So I try to buy organic because I know what goes into growing tomatoes. It's a lot of chemicals. So, and then also I've been seeing at Meijer, they've been selling, tomatoes. I like the cherry tomatoes, and a lot of those are sold. You'll see on the packaging, it'll say greenhouse grown. Those are another favorite that I like to grab because chances are the amount of chemicals are far less in the greenhouse than out in a natural environment. Anyways, all right, so we did mention a little bit about innate defenses. So what the plant body has certain physical things going along with it that helps it, just like our body does too, in the sense of with plants, they have a lot of waxy substances that's covering the surface of their stems and leaves, but also their roots. And so cutin and just waxes in general, there's lots of different types. Cutin is one, suberin is another. And so you tend to find the cutin type wax in the stems and leaves. Suberin type wax tends to be on the root surface to protect the roots. Oh, also in the Casparian strip, you have that. That's surrounding the vascular tissue. And then the periderm is part of the bark tissue of stems. And so suberin is also formed when the leaves fall off. So when the petiole of the leaf falls off the plant, you'll get the plant will make suberin to kind of plug that wound. And so very useful. And it's just part of the plant body as an innate defense. It's harder for chewing insects to break through this. It's harder for fungi and bacteria to make it through these layers. So if you think about what is it doing for the plant, certainly it helps with reducing transpiration. We've talked about that already. It makes sense to be waterproofed so you don't lose too much water out of your body, only through the stomata. It depends on the plant, how thick these waxes are. It's really influenced by where they live. If they're living in very dry environments, they have a lot of wax. If they're in a tropical rainforest, maybe not so much. It does help with pathogen invasion, but pathogens have co-evolved. They have the ability to produce enzymes that can start to break down these waxes. So this is just a co-evolutionary process where you have the plant and the pathogen co-evolving with each other and kind of figuring each other out and adapting and trying to one-up each other through a long interaction. Okay, so yes, innate defenses. We have these types of innate defenses where you can have thorns and spines, prickles, all of that. Of course, an herbivore chewing on that would find that very displeasurable. Here you're looking at calcium oxalate crystals. You looked at those under the microscope in Plant Kingdom. This is a leaf of an agave plant. And so again, you can imagine if you're chewing on that, maybe not so pleasant. And this is an interesting defense mechanism that's innate to this plant. This is the mimosa plant, where if it has insects crawling on it, it will quickly fold its leaves, and then the whole petiole kind of drops down. So the whole branch will kind of swoop down in response to the insect crawling along. And so that's a very interesting defense mechanism that they can employ regularly. Here you're looking at resins that are being produced. So that would be another idea of a kind of an innate defense. So yes, plants have some abilities that they're just kind of naturally programmed in their everyday life to have. But for what we're going to talk about, we're going to be looking at secondary metabolites. So again, pulling up this big picture idea. So we're going to be looking at what kinds of secondary metabolites are they making in response to a particular pathogen or herbivore or whatever. So going back to that wicked plants, if you wanted the Subtitle is The Weed That Killed Lincoln's Mother. I put this slide in there, and it's because of the snake root plant. So if you have dairy cows eating snake root, the milk can get contaminated with trematol from the snake root plant. And if you drink contaminated milk, you can either get sick or, in this case, Lincoln's mother died from that. So I have in the notes section a little blurb about that if you're curious. So again, it's not really that people are chewing on the plant and eating the plant. They're eating milk contaminated because the cows ate the plant and the drug went right into their milk. So something to think about in terms of, doesn't always have to be directly the plant. Okay. So I put this up here mainly just to get you thinking about the difference between constitutive versus inducible in terms of, usually when we say constitutive, that means it's present continuously. Often if it's a chemical that's being produced, it's usually at low levels, but it's pretty continuous. And then inducible means that that chemical will get synthesized in response to something that the plant has experienced in that moment. And so it's kind of on the fly response, not something that they're continually doing. And so I'm going to be focusing much more on the inducible idea, on how do the plants respond when they sense that they're getting attacked. Okay, so we often use the word pathogen as a broad term, which could mean anything that's attacking the plant. So sometimes I'll just say the word pathogen and I'm thinking broadly. And then sometimes we will be more specific and say it's an herbivore, or pest is pretty broad as well. Usually when we say pest in the plant world, we're talking about insects, usually. Microbes, usually when I talk about pathogens, because of my training, I tend to focus on microbes, the plant, the bacteria and the fungi, not really the viruses. I don't do much with viruses, but certainly they fall into that category for sure. And if you were going to rank out, fungi cause more plant diseases than bacteria, for sure. I think viruses are also, I think this is the correct ranking. Although the more they discover about viruses, that ranking could shift. But right now, so far, all the different pathogens that have been studied, there's more fungi that cause problems than bacteria or viruses. Herbivores, that can include mammals, insects. And if you just think about the insects, half of the 2 million insect species that we know of, half of them feed on plants. And they've been around with plants for about 350 million years. So that's a lot of interaction going on. Lots of co-evolutionary processes between the plants and the insects that want to eat them. So lots of interesting studies looking at how this dynamic plays out. So when you look at some of these insects that are feeding on the plant, you know, if you think about it from the insect's perspective, they're just trying to get nutrition, really. They're trying to make a living, so to speak, get enough nutrients. So there's different ways to do that. Some insects are very well adapted to just put a piercing mouth part into the phloem, vascular tissue, and suck away the sugary solution in there. And so aphids are awesome at doing that, unfortunately. And whiteflies, we have both of those in the greenhouse on a regular basis. So yeah, you got to admire them because they are exquisitely adapted to have their stylet mouth part go into the phloem and very precisely get at the sugar. And so they actually say the word or phrase precision herbivory because they're just so good at targeting one little spot of the plant. And so that stylet mouth part is just really well designed. So phloem feeders are one big category that are studied, and then cell content feeders, they're not quite so precision. In this case, we're thinking about the ones that are like busting open the cells, lysing the cells, and just sopping up all the nutrients that spill out. So mites and thrips are good for that. Because they're pretty small at low doses, it's modest damage, but of course, if their population levels skyrocket like it can in a greenhouse, you can have very much damage. And then the chewing insects are, of course, the bigger ones that you can think about, like caterpillars and beetles and grasshoppers that have very well-defined mouth parts that can chew away. So those are the ones that certainly do the most physical damage to the plant and can very quickly destroy large parts of the plant body. I just want to show something to you really quick. Beetles got me thinking. I showed this to my other class. Isn't that amazing? So this is a close-up picture of a real frog-legged leaf beetle. Isn't that amazing? So this is part of the Nikon Small World photograph competition where they have expert photographers compete. And I put the website, I can post this if anyone's interested. They have beautiful, beautiful images of all kinds of biological things. I was most entranced by the insects. I just found the detail amazing. So beetles can attack plants for sure. This is an ant face. Reminds me of the movie Alien. And so ants certainly have a long coevolutionary history with plants as well. And this one is just cool. Bold jumping spider. Look at that. That's just phenomenal. It's so beautiful. Anyways, I can post these. It's just and give you the link. It's really a good way to de-stress. Like if you just need a break from studying, there's 92 pictures and you can just click through them and they explain what they are and you can get more information on everything. It's awesome for just de-stressing. So I'll post that and you can look at those when you have time. I know it's a rough time of the semester. Okay, so let's get back into, let me share that again. Into here. Okay, so I don't know why it did that to me. Oh, come on. Hang on, my computer does get confused if I switch PowerPoints. Oh boy. Let me do it this way. Okay, now it looks happier. And share again. Okay. Here we're looking specifically at nematodes. So not all insects are evil for the plant, here we're showing, yeah, but just remember there's plenty of good insects out there that I'll mention a few cases, but we're kind of focusing on the negative ones that are attacking. Here is a case of nematodes. There are good nematodes out there that can be used as biocontrol agents, but here you're showing the case of the ones that are causing I don't know, if it's soybean cyst disease maybe is the soybean plant they're looking at here. They're called cyst nematodes because they form these cyst-like structures. And so again, if you invade the plant roots and start damaging the tissue, obviously those roots aren't going to be functioning very well as they should for the plant. And so nematodes can cause a lot of damage on a lot of different plants, especially some really important crop plants like soybeans. So you gotta wonder, okay, so the plant's stuck in wherever it landed, wherever the seed landed and germinated and grew, it's stuck. That's where it has to live its life. And it's assessing its environment constantly and having to respond appropriately. And so lots of different pathogens and pests will be attacking the plant. And so it makes sense that it would have certain secondary metabolites that would be very effective against this pathogen, but maybe not so much against that pathogen. And so they started looking into this idea. It's been ongoing research for decades now. And they do realize that, yes, plants are definitely able to pick up on who is attacking them and then respond appropriately to that particular pathogen or pest. And so that's the induced idea, where it has to wait to get attacked and figure it out quickly and then try to mount a response very quickly. And so from a plant's perspective, if you wait, that way you don't have to make all of these secondary metabolites all the time. That would be incredibly draining for the plant to always have everything up and running. It's just, that would be too much of a drain on its resources. So it does make sense that it would hold off and just wait to see what's going to happen and then respond to that particular pathogen. The downside is you got to be very quick. You got to be very capable of sensing who's attacking you. And then you have to be very quick in your response. So usually the gene expression patterns have to shift very quickly. You have to have very quick gene expression going on, transcription and translation, and then get everything where it needs to get to in a big plant, if you think about how, especially large plants. So we use this term, elicitors. Elicitors are chemicals which trigger a defense response. So this is a broad umbrella-like term that we use, elicitors. And so there's all kinds of different elicitors that fit into this category. Here's one example, HAMPs. They just say the acronym HAMPs, which are herbivore associated molecular patterns. Big long mouthful. Luckily, we can abbreviate that down to HAMPs. So we'll talk more about HAMPs, and then there's MAMPs and things like that. So we'll get into these as we work through this. But basically, if you think about, okay, there's some kind of chemical coming from whatever's attacking the plant. It's usually, maybe it's part of the, let's say it's a fungus, maybe it's part of the fungal cell wall or the bacterial cell wall, or maybe it's something in the mouth part of the insect that's chewing on the plant. But whatever it is, when the elicitor is recognized by the plant, there's tends to be some common responses. You do see often kind of a burst in calcium levels. And remember, calciums are second messengers. So that does make sense that the plant would ramp up calcium because that can be used in a signal cascade to affect gene expression. So it makes sense that they would have these upswings in calcium. You also get the calmodulin proteins, very active. Those are the proteins that bind to calcium, and then that complex will usually impact gene expression. And so often you see what they call calcium-dependent protein kinases. Kinases are just enzymes that transfer phosphate groups, their job. And so again, this is dealing with a signal cascade. We know that phosphate transfer is a way to send a signal in a signal cascade. And so this is just, all of this is talking about ways to ramp up the signal cascade system. And so that you can effectively get gene expression patterns shifting. So downstream targets, usually it's a phosphorylation transfer from one protein to the next, and then eventually that can impact gene expression. So basically, these elicitors kick off second messengers and all of the associated molecules that go with that so that you can ramp up your signal transduction and get the right genes turned on in response. You also tend to see key hormones getting made or released from being conjugated so that they can become active. And we'll talk about that as well. And then MAPK is an acronym that stands for mitogen activated protein kinases. They named that when they discovered it in the animal world, but actually terrestrial plants have the highest number of MAPK genes than any other organism. So these have been heavily studied in animals, but it's actually plants win. They have way more of these protein kinases. So protein kinases, again, involved in transferring phosphate groups. So again, it's just part of the signal cascade process that the plants will be using. All right, so the next thing we're going to do is get into some examples more specifically to show you like what kind of hormones are involved and what is exactly going on. But since that's going to take a bit, we'll stop here and we'll do that next week. So we have the exam on Friday. Speaking of the exam, so I have a pretty solid draft, multiple choice, and then I have drag and drop and essay as well. So you'll see it like multiple choice, true, false, fill in the blank, matching will be in one. It'll be in the multiple choice section, and then I have a essay, drag and drop in the other section. So you'll, again, it's 2 versions of the exam. Work through one, submit it, and then you can get into the other one if you remember to totally X out of lockdown browser and all of that, and go back into the second exam. Okay, so I will get here as close as I can to 10 to the hour on Friday, and we will make sure that your computer is fully charged and all of that, but it's this kind of the same basic format. You'll have 2 versions of the exam to work through. And please ask questions during the exam. If you're not sure what I'm asking for, please ask, okay? Really ask, because I don't want you to go down the wrong path, all right? I will nudge you in the right direction, okay? 2 essay questions. Yes, 2 essay questions and then some drag and drop too.